paper 2, A-level biology. So let's get started. The first question here, heat stress is a condition that often occurs in plants exposed to high temperatures. High temperatures are really important here. And heat stress is a major factor in limiting the rate of photosynthesis. So heat stress decreases the light-dependent reaction of photosynthesis. So when a question like this comes up, you need to think what are the products of light-dependent reaction here. So we've got ATP and reduced NADP. So we need to explain why this leads to a decrease in light-independent reaction. So again, you should remember that products from light-dependent reaction are needed for light-independent reaction. So how does that reduce? Because of the less light-dependent reaction means that we've got less ATP and less reduced NADP. So that's our answer. Next question, another effect of heat stress is a decrease in the activity of enzyme Rubisco. So Rubisco is the enzyme involved in the light-independent reaction in the first stage when 5-carbon molecule combines with carbon dioxide. And the decrease in the activity of an enzyme means that the rate of the reaction it catalyzes becomes slower. Then we've got a statement that a decrease in the activity of enzyme Rubisco would limit the rate of photosynthesis. What part of photosynthesis is Rubisco involved in? Light-independent reaction. So how does that limit? Because less carbon dioxide will react with IUBP, so that 5-carbon molecule, so less GP will be produced. Next question, where exactly can you find Rubisco? In the stroma, because this is where light-independent reaction takes place. Then we've got the question with the graphs. So the scientists investigated the effect of temperature on the activity of two enzymes isolated from the leaf cells of cotton plants. And we've got Rubisco and Rubisco activase, and the job of this enzyme is to activate Rubisco. And we've got two figures here. Label them, make sure that you know what is happening. So Rubisco activity against temperature, we can see that this increases when the temperature increases, and the Rubisco activase activity increases up to certain point here, around 25 degrees, and then decreases. So, but what's the job of the Rubisco activase? It's of course to activate Rubisco. So we can see that even so, after this 25 degrees, Rubisco activase decreases, but Rubisco activity still increases. So that's quite important information. So the scientists concluded that, and only this statement is in your interest. So heat stress reduces the activity of Rubisco in plant leaves by affecting Rubisco activase, and to use that information to evaluate that conclusion. So here they don't say to support or so to negate the conclusion. You need to think about both factors, yes, no, and why. So for what we can say is that Rubisco activity increases with temperature. Correct. Rubisco activase activity decreases at high temperatures above 25 degrees. We can see that here. And the results suggest that activase does not affect activity of Rubisco. That's correct. So that's what we say, that even so this decreases after 25 degrees, the Rubisco activity still goes up, still increases. And another point that we could say here, that the results are only for cotton. Why do we know it? Because it says that the enzymes were isolated and they are only of the cotton plants. And then we got our 4 marks. Next question, it's about the mutations. So we need to put a tick in the box next to statement which describes incorrectly the effect of the mutation in an exon. What's exon? It's a non-coding sequence. So exon, sorry, is coding sequence. So the true a substitution might not result in the change to the encoded amino acid. True, false. and inversion will result in a change in the number of DNA bases. That's incorrect, because what happens here, inversion changes just the way the bases will be arranged in the polynucleotide. And then we've got deletion results in a frame shift, which is correct, and the addition will result in a frame shift, which is correct as well. So what we've got here, I got to describe how alterations of to tumor suppressor gene can lead to the development of tumors. So here it's a general question, because they didn't tell you what kind of methylation or acetylation are you looking at. So how the tumor suppressor genes will lead to tumor only if you increase methylation. Okay, so increased methylation, it's going to cause mutation in tumor suppressor genes, because the gene is not going to be expressed. And what's the normal function of the tumor suppressor gene is to, of course, be expressed, because they are stopping that cell division. So if they are not expressed, that will result in uncontrolled cell division. So next we've got the type of malignant tumor, and we've got information that divides every 8 hours, so that means 3 times per day. So starting with one cell, how many tumor cells will be present after 4 weeks, and we've got to work it out in a standard form. So what I did here, firstly work out how many days is in 4 weeks, so that's 28 days. Multiply this by 3, because this is how many times per day it's going to divide. So we've got 84 times. And to work out the number of cells, it's a formula 2 to power n, where the n is number of divisions, so 2 to power 84. And that gives us the answer 1.93 times 10 to 25. Right, so here we've got aerobic and anaerobic respiration in the yeast, and the students use the equipment shown in figure 3 to measure the rate of respiration. So what the student did, she put the flask in the water bath, so the yeast culture reached a constant temperature, and then left the equipment for 1 hour before starting investigation. What we need to remember here is the fact that you need to think what's in the equipment given. So we've got the yeast culture in glucose, okay, and we've got the alkaline here solution that will absorb oxygen. So what do we need those? Of course, we need them for aerobic respiration, and here we've got the liquid. So you should remember that every time the liquid has moved, okay, we then need to be thinking about the volume or the pressure in this situation here. So question is to suggest one reason why it was important that the students left it for 1 hour after the yeast culture reached a constant temperature. So why are we leaving them? Let's have a look where they are. They are here in that solution. What's in them? Oxygen. So the oxygen, it's used in the respiration. Okay, so that's the reason. Then we've got investigation and colored liquid move to the right. So if we move to the right, so let's go back to the picture. Okay, so it moves that way to the right, means that the volume, okay, here increases. So as I said before, okay, thinking why does it increase? What could possibly be produced? Okay, so what are the products? Okay, so products of anaerobic aerobic respiration, sorry, anaerobic respiration produces carbon dioxide, so that increases the volume of the gas. Then we had the mass calculation here. So we've got the liquid that moved by 1.5 centimeters in 24 hours. So this is our length, and the diameter was given. And you need to work out the volume of gas being produced per that unit of per those units. So good we've got a formula, so we write down everything what we know. We know what's pi. The length is 1.5. The diameter is 1 millimeter, so we convert the unit, so that's 0.1 centimeters, and we can work out the radius from here, which is 0.05. Using the formula, okay, we're working out the volume, so that's the volume in 24 hours, because that's what was in the question, in 24 hours. So to work out per hour, we need to divide this volume by 24. So the final answer is 4.9 times 10 to minus 4. Okay. So here we've got the question that shows us then the growth curve. And what they're asking you, why the log scale, explain why a log scale was used. So that's just AO1, so, well, AO2. So the large range of the numbers was here in this investigation. That's why we're using that. And the many cells die. So if they died, that means, of course, there was no respiration. So suggest one reason why. So why was there no respiration? Because there was no glucose for respiration, for example. Right? Here we had the maths question. So again, we've got lots of information here, and the idea of this is to then match them. So reading through the question, population size, so that's 2000, okay. R is a growth rate, which we've got here from 0.5. T is a time, so that's 10 hours, and we've got E from here. So we need to put in the calculator using the formula provided is just this, okay. So 2000 times 2.72 and to the power that we've got given in there. So I didn't write the answer here, but if you put this in the calculator now, you will get a correct answer. So question number 4 is about the succession, and they measure the percentage cover of different species. Okay, well, we've got those different species, so we've got it here. We've got a key for those at different age. Age is in here, and the percentage cover just there. So what we need to do, we need to describe the method, okay, so give the method how you would determine the mean percentage cover for beach grass on the sand dune. So the method, how can you do it? Okay, so make sure it's something that people can follow and do really easily. Okay, so clear instructions, so determine the position randomly. Okay, so for example, using the random number generator, make sure you've got the large number of quadrants and divide total percentage by number of quadrants. Where is this answer coming from? From the mean. Okay, so be careful. There's lots of questions like that asking about the mean. So make sure you include that in your answer as well. So then we've got the scientists conclude that the results shown in figure 5, so use that figure 5, were due to succession taking place, and using the figure 5 explain how. So coming back to figure 5, okay, you need to of course know your content, so you need to know what the succession is. So what are the pioneer species, what's the function of them, that they make the conditions less hostile, then talk about a climax community and the species found in the climax community. How are you going to find out the pioneer species are the ones that comes up first? Okay, so that's where is our answer then coming from, that the beach grass is the pioneer species. They change the abiotic environment, make it less hostile for, and then come with the name of the species that comes up after. So for example, conifer trees, and those conifer and hardwood trees will be present in the climax community. So that's our using information explaining how the succession could take place in this new situation here. So then we've got another investigation, how the proportion of sunlight reaching the ground change during succession. So some of the results are obtained and put in on the graph here. So we've got the percentage of full sunlight, okay, and then we've got the age. So using that figure 5 and 6, what we just looked at, you need to explain the results. So as you can see here, the sunlight goes down from 100% to like 25, let's say. So what happens here, obviously, how is it possible? Because the trees are blocking the sunlight. So using the evidence from figure 6, what can you conclude about the net primary productivity? Okay, in the sand dunes that are older than 1,000 years. So what we can see here, above this 1,000 years, that we've got the same constant, percentage of the sunlight. So what is the net primary productivity? Of course, will stay constant because photosynthesis and respirations are constant through this. It's constant supply of the sunlight, hence the answer. Right, so we're going for question 5 now. So we've got the information here about the drugs, and the function of those is to remove excess fluid from the body, and the scientists investigated the effect of those drugs on the volume of urine. So what we're thinking here is if there is lots of water in the urine, if you are not, the volume will be high. If there is less water, the volume, of course, will be less. And then you've got 3 groups of the people here, A, B, and C. So we've got information here, what they actually were given. Okay, so group A, a tablet, group B, different tablets, and the C were giving the placebo, so a tablet that did not contain any of the drugs. And there were investigation taking place. The results shown in the figure here, giving you standard deviation. Standard deviation, it's important. Every time you see it, make sure you write a comment next to the table or the graph. So here you can see that the mean volume of the urine is produced, and you've got the results for each group. So looking at the standard deviation, what you can conclude, they don't overlap. So if they don't overlap, that means there will be a significant difference between those results. So coming for the question is that all volunteers were given the same food for 3 hours and suggest and explain so why they were given the same food. Okay, so why the same food? Because what we're looking at the excess fluid. Okay, excess fluid. So if we're giving the same food, we are not going to affect the water potential of blood, and also that affects the volume of urine produced. So that's why, according to the investigation. So using the table 1, including of course the standard deviation, conclude, so say what you can see about the effectiveness of those drugs. So what we can see, that both of them are of course more effective than placebo, so that was the tablet without any of the drugs. And what we say using the table, okay, there is a significant difference what we say because standard deviation do not overlap. So we've got the drug that is sometimes used to treat high blood pressure, okay, and suggest how this drug could cause a decrease in the blood pressure. So that will then of course lower the volume of the blood, okay, because got the high blood pressure. High blood pressure means lower volume. That's where the answer is coming from. 5.4, we got the drug that will inhibit the absorption of sodium and chloride ions from the filtrate, okay, and explain how this drug causes an increase in the volume of urine. Increase in the volume means more water in there. So water potential of the filtrate will decrease. So if that increases, of course, we will have more water in the urine, so less water will be reabsorbed by osmosis from the filtrate. So and we know that osmosis is the one that takes place in the collecting duct. So increasing volume means more water in volume, so the water potential in the filtrate must be lower. Right, so again we've got the table with the information here, the mean rate of flow of blood plasma into the kidney, And we need to be then looking at the maths question again. So the mean rate of flow of blood plasma is 60% of the mean rate of blood flow into the kidney. So how much greater the flow of blood into the kidney with one of the drugs and then with group C, okay, after 4 hours, and we need to give the answer in centimeters cubed. So what do? We're looking at those two. We take them away. So we've got 85 as the difference because that's what they want. And we then multiply this, okay, by 4 hours, and 60 seconds is in an hour. So 20,400 refers to only 60%. That's what the question says. So doing the proportion, we're looking for 100%. So the answer, final answer, will be 34,000 centimeters cubed. Okay, so that's our answer. Right, question 6, genetic crosses. So the observed ratio is not always the same as expected. Say why, so give the reason for this. So of course, due to random fertilization of gametes, and for example, the sample used would be small. Here we've got the long pathway about the cross is about the tomato plants and the genes for height and for the type of the leaf on the same homologous pair, so that means that they are linked, okay? And then we've got those alleles described here. So rather than reading through this again and again, I've just wrote this down on the actual figure. So dominant, it's tall, recessive, it's the other one, normal, big M, and the mottled lower M, so that's recessive, that's dominant. So then I don't need to read through this all over again, saving myself time. So the biologists carry out the crosses, okay, between plants heterozygous for both genes, and those are the results. So before I look through anything, I then look, right, so I've got huge amount of those, are quite low in net. So I just write this down here. I've analyzed the table, and what would be the genotype of that plant? So look, according to my writing here, both characteristics are recessive, hence the genotype. Use the information provided and the results in table 3 to explain those. So as we say, the genes are linked because they are on the same excuse me, they're on the same homologous pair. So if they are linked, of course, we can then see that there will be less of those offspring. We've seen that in the table that we've labeled. So that's due to being linked, and that, for example, crossing over has occurred because we've got some of those. Okay, we've got a small number, but we've got it. So that's crossing over. And then complete the table 4 to show the ratios if they were on different homologous pairs. So if they're on different homologous pairs, that means that they are not linked. So it's a normal dihybrid cross. So you're doing a dihybrid cross for the 2 heterozygous parents, and you know that the ratio for this would be 9:3 to 3:1. Okay, so normal dihybrid cross for the 2 heterozygous parents. At the end, you're gonna get that. Question number 7, neurotransmitters released in some synapses in the brain. And we've got the dopamine, which is similar to acetylcholine. And dopamine stimulates the production of nerve impulses in postsynaptic neurons. Describe how. So what's the process of this in the postsynaptic neurons? And here they told you not to include do not include in your answer events leading to the release. Okay, just say what happens after. So what happens after? Of course, dopamine will diffuse across synapse, attached to the receptors on the postsynaptic membrane, and here make sure you say membrane, and stimulates entry of sodium ions and leads to depolarization or action potential. Only of those will be fine. So that's a nice friendly question here. 7.2. So we've got that dopamine has a role in many processes in the brain, including pain relief, and the release of this can be stimulated by chemicals. And endorphins will attach, okay, to the receptors, and we need to explain how, using the information that morphine is a drug that has a similar structure. So that means what? How? Okay, so the morphine will attach to the receptors, and more morphine will be released to provide pain relief, which we can see it from the question here. Right, GABA. Many questions you've probably seen with GABA before. So neurotransmitters, and they will inhibit the synapse, and GABA causes negatively charged, okay, chloride ions to enter postsynaptic neurons. So what does that mean? That they will be leading to hyperpolarization. Explain how will that inhibit. So inside of the postsynaptic neuron becomes more negative. So how can we overcome this? By getting more sodium ions to reach the threshold for the polarization to take place. Right, genome definition, all the DNA in the cell, and here we've got a figure that shows you the information about the transcription factors. What's the job of transcription factor? Is to start transcription, and of course gene needs to be expressed. So figure 8, okay, we've got here chemicals bind, breakdown of DNA into small fragments and add antibodies specific to this transcription factor, removed DNA, okay, so, and then finally we've got the transcription factor involved. So what the question's asking for, explain why the antibody binds to the transcription factor, okay, so what's the antibody? Of course, it's a protein, so specific. So transcription factor will initiate the transcription. So what we've got here, so the transcription factor has a specific tertiary structure, and they've got the complementary shape. So that's why they can bind together. Then we've got the figure 8 to use and say what the DNA will be made of, okay? So the DNA that we're using. So if you read through this figure again, there will be DNA, transcription factor, and antibody, okay? So what we've got that the DNA, we've got it in there. So let's have a look. What was that? Transcription factor was antibody here, and DNA. So the answer comes from just analyzing the flow chart here. So we've got the soybeans now, and we've got information here that they contain protein known as P34 that causes genetic allergic response, and scientists have created transgenic soybeans that produce single-stranded cDNA. Okay, so now you're thinking section 8, how can you make the cDNA? And that cDNA will prevent transcription of that gene. Okay, and they use recombinant plasmid, so the plasmid that has the DNA of your own interest and plasmid's DNA to transform soya bean cells. And the question is to suggest how single-stranded cDNA could prevent. Well, single-stranded as the term stands, okay, it's single, so it can bind to the gene, and doing so, they will prevent the transcription, so it cannot act as a template for the transcription to take place. So describe the role of two enzymes involved in the insertion of DNA into plasmid. So we need to cut plasmid firstly, so that's using restriction endonuclease, and we need to then join those two DNAs with the enzyme which is called DNA ligase, so that joins gene to plasmid. Right, so soybeans were screened for the presence of the protein, and this process will involve gel electrophoresis to separate. So just how can we then separate those? So how can we separate? By size, by mass, by length, by charge? Okay, so what's the length, the number of amino acids, charge? Okay, that's to do with the R groups as well. So that's where the answer is coming from. Lactose then. Okay, we've got a long pathway here about the lactose, which is a sugar in the milk and is hydrolyzed by enzyme lactase, and it's a lactase enzyme is essential for to newborn mammals, as milk is the only source of food. So most mammals stop producing lactase when they start feeding on other food sources, and humans are an exception to this because some continue to produce lactase as adults. And the ability to continue producing lactase is known as LP, okay, and it's controlled by dominant allele. That's important. And the number of hypotheses based on different selection pressures have been put forward to explain this. So dominant allele, capital letter, selection pressure, that will then lead to, of course, we need to adapt due to different selection pressures. And the one hypothesis for LP in humans suggests that selective pressure was related to some human populations, okay, and describe how. So we continue to produce this. So what does that mean? What's the reason? The reason of this is due to mutation, and obviously we know that milk provides nutrients, and the individuals are more likely to survive and reproduce because they're dealing with those selection pressures. So it's a directional selection, okay, to adapt towards the specific aspect, so the frequency of that allele will then increase. So use the information provided, explain why the number of people showing LP would rapidly increase, okay, once selection for this condition has been established. Why? Because we read through this pathway before and we know that it's a dominant allele. What's the definition of the dominant allele? It's the one that is always expressed when present in phenotype. Right, next question, lactase disease is caused by the mutation in DNA, and this mutation does not occur in the gene coding for lactase enzyme. So not in the gene, okay, so where else it can be? It can be, for example, in the promoter, okay, so mutation in the promoter gene for that transcription factor, and the lactase gene will continue to be transcribed. So that was a bit, that could be a hard question to think of about the answer, but promoter is something that we should be thinking about in here. Finally, we've got our last question, question 10. There's a lot to read in that question, so I will let you read through that question, have a look at my labels here, and we will start in a second. Right, so what I've labeled here, rod cells, what do we need to know? That they only allow you to see in black and white. There are 3 neurons attached to one bipolar neuron, cone cells, color 1-to-1, autosomal recessive allele. Okay, so that means they're not sex-linked, so only with looking at the recessive alleles, sex-linked. And then we've got the man and the woman idea here. It's the fact that the man has only one X chromosome. Advantages, so what's better? So looking at the questions, people with complete disease will have difficulty in seeing detail. Why? You're using line 2 to 3, so the information about rod and cone cells, and this is what we need to then recall. So no cones, only rod cells will be present, and the idea that cones are connected to a single neuron and they can separate sets of the impulses to brain, while the others cannot. Right? So Heidelberg equation here to use. So 10% of population is affected. So 10% of the population is affected, and we know it's a recessive disease. So what we've got here, so our Q square equals 10%, so 0.1. Using the equation, I can square root it, so I can get my Q. So 0.32, it's a Q. Using the equation P plus Q equals 1, I can work out my P, so 0.68. And the heterozygous, it's 2PQ, so I multiply those by 2, and the answer is 43%. Right? Then we've got the red-green color blindness affects more men than women. So we did discuss that, so sex-linked. So those are all the possible genotypes for the woman. Those are for the men. Why? Because that gene is on the X chromosomes and the females requires 2 of those alleles here to be present, okay? And they can also be heterozygous, okay, but male obviously cannot, and they only required 1 allele for the disease to be present. Right? So next part of this question, so people with the red green color blindness are unable to distinguish between red and green, and also between other colors. So why? So we then recalling our knowledge about the sensitivity of those cells. So the green sensitive pigment cones that detect green light are non-functional, what we've read about in this long pathway, and 3 different types of the pigment are here. We also got 3 different types of the cone cells, so different colors can be seen due to the stimulation of more pigment. So current research into the treatment of red-green color blindness involves the use of induced pluripotent stem cells. So give the reason how they could correct the red-green color blindness, so improve, so we can see. So what are the possible answers here? It's the fact that those induced pluripotent cells will divide, and what they can do, they can develop into different type of the cells. So in here they will develop into cone cells, which will allow them to see in those conditions. And the last question we've got here is to use the use of induced pluripotent cells could have advantages over gene therapy. Okay, and use the information as suggests, so give the reason why. So what will be the answer here? It's the fact that they have a long-term effects. There is less chance of rejection of the pluripotent cells. It's only a single treatment, and the side effects side effects from using viruses in the gene therapy, we don't need to do that using pluripotent stem cells. Right, so that's the whole feedback on paper 2. I hope you've got now a clear vision of the working outs and the answers, and make sure that you revise your content and do lots of questions using the approach of use the information, conclude, evaluate the conclusion. That seems to be a pattern in 2018. Right, so see you later.